Good afternoon to all the EAPCET aspirants. This is your Prasanna ma'am from NEET WORLD. So yes, we are in the chapter Plant Kingdom. Yesterday we have finished what ma? The types of classification of plant kingdom. That are three types of classification, majorly artificial system of classification, natural system of classification and phylogenetic system of classification. Post that we have also looked into branches of taxonomy, three branches of taxonomy. First is numerical taxonomy or phonetics, cytotaxonomy and chemotaxonomy. After taxonomy, we have started dealing with 5 divisions of plant kingdom. First is division algae is done, and today we are going to deal with bryophytes and pteridophytes. In our next class, we will be finishing off gymnosperms and angiosperms. Post that, we will be exercising all the EAPCET PYQs. So, without much delay, let us get started. First, we will finish off the general characteristics of bryophytes. Bryophytes are most commonly known as amphibians of plant kingdom. They are most commonly known as amphibians of plant kingdoms, and they are the primitive land plants, and they are the first non-vascular cryptogams. They are the non-vascular cryptogams. Non-vascular cryptogams. Fine. They are most popularly known as what? Amphibians of plant kingdom, primitive land plants, and they are non-vascular cryptogams. They are archegoniate, embryophytic, atracheophytic cryptogams. They are non-vascular terrestrial cryptogams. These are the synonyms of bryophytes to be remembered. You will be asked with the questions of this by using any of the synonyms. Okay, and these are the first embryophytes. What are the first embryophytes in plant kingdom? Bryophytes are the first embryophytes in the plant kingdom, and they are the first archegoniates also. Archegoniate, embryophytic, atracheophytic cryptogams. Okay, atracheophytic, non-vascular terrestrial cryptogams. Remember the terms carefully. When we have to look into the habitat of bryophytes, they are mostly grown in the shaded areas like on hills. Damp, humid, shaded localities, bryophytes are found. If you see the habit of this, you do not find true root, leaf and stem, but then you can find root like, leaf like and stem like structures. Root like rhizoids, leaf like phylloids and stem like colloids. Okay, root like rhizoids, leaf like phylloids and stem like colloids. Rhizoids can be unicellular or multicellular. Unicellular rhizoids you can see in liverworts. Multicellular rhizoids you can see in mosses. Fine ma, fine with the general characteristics. Any doubt anywhere? Just take a screenshot of this if you want. Very important slide beta. And then plant body is more differentiated than algae, but it does not have true root, stem and leaf. It has root like, stem like, leaf like structures. Root like what beta? Rhizoids, stem like colloids and leaf like phylloids. Here if you see the gametophyte, gametophyte is haploid and gametophyte is the main plant body in the bryophytes. Okay, gametophyte is the main plant body in bryophytes. They have multicellular sex organs. They exhibit multicellular sex organs, antheridium and archegonium. Antheridium is multicellular and it is jacketed. Okay, it is multicellular and jacketed. So, it is jacketed. Inside antheridium, you have androcytes or spore mother cells. Androcytes or spore mother cells undergoes mitotic division, mitosis. Okay, these are the gametophytic structures, right ma? Gametophyte is haploid. Antheridium is haploid here. Archegonium is also haploid. Remember that. Okay, and this is covered by a jacket, sterile jacketed sex organ. Okay, inside the antheridium you have androcytes or sperm mother cells. These are what? Sperm mother cells. Sperm mother cells. Okay, sperm mother cells, they undergo mitosis and they release 2 anthrozoids. Anthrozoids have flagella and they exhibit zooidogamous oogamy. Look into the gametophytic structure of Archegonium. Archegonia here has a long neck and venter, inside which you can see neck canal cells, venter canal cells, and at the bottom you can find the egg cell. Understood ma? It has a neck and a venter. Neck, below the neck you can see neck canal cells, venter canal cells, and at the last basal region you can find out the oosphere or egg cell. Egg cell is also called as what ma? Oosphere. The antherozoid, which is biflagellated, undergoes zooidogamous oogamy. It swims across the water, zooidogamous oogamy, and this antherozoid will fertilize with the egg, which is oosphere. Clear, right? Any doubt here? Two gametophytic structures, one is the male gametophyte and the other is the female gametophyte. Okay, antherozoids exhibit what type of oogamy ma? Zooidogamous oogamy. These are called as amphibians of plant kingdom because they are dependent on water for fertilization. Okay, of course they are in the terrestrial habitat only, but then they are dependent on water for fertilization. That is why they are called as what? Amphibians of plant kingdom. Next you see ma, these are the site of fertilization. What is the site of fertilization? Archegonium, because inside archegonium only you have oosphere or egg cell. This biflagellated antherozoid travels all across the neck and reaches the egg or oosphere. So, the site of fertilization is what? Archegonium. After fertilization of haploid antherozoid fertilizing with haploid egg or oosphere, what you will get? A diploid zygote. Haploid egg or oosphere results in the product of what? Post fertilization, diploid zygote is formed. Okay, diploid zygote which matures to diploid embryo. First embryophytes are what? Bryophytes. This embryo develop into what? Sporophyte. Okay, so zygote is diploid, embryo is diploid, and sporophyte is also diploid. Now you look into the sporophytic body. Okay, of course gametophytic plant body is the major plant body of bryophytes, and next it is what? Sporophyte. Sporophyte is attached to photosynthetic gametophyte for nourishment. Okay, sporophyte lives as a parasite, partial parasite on the gametophyte over here. This is dependent on the gametophyte for nourishment. Spore mother cells, here in the sporophyte, spore mother cells are present. Spore mother cells are diploid. This undergoes reductional division or meiosis, produces 4 haploid spores. 4 haploid spores, and all spores are alike. So, you call it as homosporous condition. Only homosporous condition is observed in bryophytes. I hope this is clear beta. What are the gametophytic structures? Over the gametophyte you can find antheridium and archegonium. Inside the antheridium you have androcytes or sperm mother cells. They are haploid, undergoes mitosis, produces haploid antherozoids. They exhibit zooidogamous oogamy, fertilizing the egg which is at the basal region of archegonium, and here the archegonium is the site of fertilization. Post fertilization you are getting what? Diploid zygote. This diploid zygote matures to diploid embryo. Diploid embryo, these are the first embryophytes, and then this matures to diploid sporophyte. Inside the diploid sporophyte you have diploid spore mother cells. Spore mother cell undergoes what? Meiosis or reductional division to produce 4 haploid spores. All spores are alike. So, these are what? Homosporous. Clear, right ma? Now you see this spores will develop into gametophyte. Spores landing onto the suitable substratum will develop into gametophyte. It is thalloid gametophyte in the liverworts. In liverworts it is thalloid gametophyte, and this thalloid gametophyte which you see in the liverworts, and then in mosses if you see, spore will first develop into primary gametophytic structure called as protonema. Spore, if it is a liverwort, this develops into what ma? Thalloid gametophyte. If it is maturing into a moss, this first turns into a primary gametophytic structure. That primary gametophytic structure is primary gametophyte, that is protonema in bryophytes. Very important, remember the life cycle stages are asked not just in EAPCET, even in your NEET examination. If you have to consider NEET 2025 paper, you got 2 questions only on the life cycle stages of bryophytes and pteridophytes. 2 multi-statement questions are asked from the same chapter, bryophytes and pteridophytes. That important it is. So please listen with complete focus and concentration. If you are having any doubt, please drop a message in the chat box. I will be answering your doubts. Okay. So here I hope this is clear. Haploid spores develop into a thalloid gametophyte in the liverworts and into a primary gametophyte structure, that is protonema, in mosses. And then you see bryophytes are classified into 3 classes. First class is what? Hepaticopsida, second is Anthoceratopsida, third is Bryopsida. Okay. So first Hepaticopsida, best example is what? Liverworts. Anthoceratopsida example is hornworts. Bryopsida example is what? Mosses. Clear, right? Hepaticopsida liverworts, Anthoceratopsida hornworts, Bryopsida mosses. And then you see first let us learn in detail about liverworts. Liverworts habitat is same as that of bryophytes, moist shady places like banks of streams, marshy ground, damp soil, bark of trees and deep woods. Of course, this is very simple, but then you will get confused when it is asked in the form of statements. Need not to confuse at all. Here you just remember shady and dampy places are the habitats of liverworts. Next, habit. Plant body is thalloid, just a thalloid plant body which is little differentiated when compared to algae. That's it. Best example is Markantia, but you can also take the example like Riccia and Porella. Fine. Now let us see the thallus of Markantia. Thallus of Markantia, if you have to observe, this is dorsiventral, closely appressed to the substratum, and this is dichotomously branched like this, dichotomously branched. In algae also you can see dichotomous branching in one algae, that is brown algae dictyota. Brown algae dictyota, you can see dichotomous branching. Dichotomous branching. Clear, ma? Just a comparative analysis, that's it. So here Markantia body is dorsiventral, this is closely appressed to the substratum, and this is dichotomously branched. And if you have to consider Porella, Porella will have leafy members. These are leaf-like appendages. Porella are what? Leaf-like appendages. They are in 2 rows on the stem-like structures. Porella have leaf-like appendages. They are present in the 2 rows on the stem-like appendages. This is the description of what? Markantia and Porella. Markantia, very important to remember, ma. This is unisexual or dioecious. Rather than calling unisexual, you can also consider this as heterothallic, wherein the Antheridia and Archegonia are present on the 2 separate plant bodies. So here you can see Antheridiophore and Archegoniophore. How you can identify? If at the tip of the Archegonia or Antheridiophore, if you can see more invaginations like this, then it is called as Archegoniophore. Consider if 2 diagrams are given and in one diagram at the tip there are much deeper invaginations, then you can fix it like Archegoniophore, and other peripheral invaginations if you can see, that is called as Antheridiophore. Antheridiophore, Archegoniophore is clear, right? So remember, Markantial structural description is asked n number of times in your EAPCET paper beta. Remember, it is a dorsiventral structure closely appressed to the substratum, okay, and it is dichotomously branched. It has unicellular rhizoids, okay, remember it has unicellular rhizoids, fine, and it is dioecious heterothallic. Both the sex organs are present in 2 separate thallus. and it has gyma cups which promotes vegetative propagation or asexual reproduction. Clear, right? So this is about Marchantia, remember it. Vegetative reproduction in Marchantia, if you see, it is by fragmentation. Most important and unique feature, that is having gyma. How can you describe gyma as? Gyma are present inside the gyma cups. Gyma are well described as green multicellular asexual buds. Here itself I am telling you, gyma in Marchantia, protonema in mosses, prothallus of pteridophytes. Remember the statement based description, that single statements which are given in your text, that are very important beta. Directly from that statements you will get the questions. Okay, remember that description properly for gyma, protonema and prothallus. Okay, so gyma here, this is the green multicellular asexual buds. This develops on the small receptacles called as gyma cups. They develop on the small receptacles called as gyma cups. Mature gyma will detach from the parent body and they germinate into the new individual. Mature gyma detach from the parent body and germinates into a new individual. Clear, right? Any doubt anywhere beta? So here you can see gyma over the thallus, here you can see gyma cups. On vegetative reproduction, that produces into a new thallus. One gyma gives rise to two thalli. Okay, one gyma gives rise to two thallus. Remember this. Okay, next. Yeah, here sexual reproduction. I already told you Marchantia is dioecious and heterothallic. It has male and female sex organs present separately on two different plant bodies, and homothallic monoecious is Rhizia. Rhizia is homothallic and monoecious. So here if you see Marchantia, Marchantia has antheridiophore and archegoniophore. Antheridiophore, here antheridiophore will release out the antherozoids which are biflagellated, as we have discussed. These antherozoids are easily dispersed through elaters. Elaters here, they are present for effective spore dispersal. Okay, they are present over the antheridiophore for effective spore dispersal. What are the structures present on the antheridiophore which are helpful for the easy dispersal of antheridia or antherozoids? If that is the question, your answer should be elaters. Elaters are present in the marchantial antheridiophore, and this antherozoid will reach the egg cell which is present in the archegonium. Okay, in the archegoniophore you will have archegonium. Inside the archegonium, again the same structure, egg is present at the basal region. This antherozoid will swim across the water and it will reach the egg which is present at the basal region of the archegonia. Okay, so what is the site of fertilization here again? Archegonium. Archegonium which is present in the archegoniophore, that is the site of fertilization again. So here the haploid antherozoid will fertilize with haploid egg or oosphere. Egg is also termed as what, Nana? Oosphere, and forms the product. What is the product of fertilization? Remember the terms carefully. Site or location of fertilization and product of fertilization is very important. Site of fertilization is archegonium and product of fertilization is zygote. Zygote is diploid. This matures to diploid embryo and diploid embryo matures to a diploid sporophyte. Clear, right? Clear till here, ma? Any doubt anywhere? Yeah. Now we will see hornworts. Hornworts are thalloid consisting rhizoids. Here you can see just, capsule which contains spores, the sporophyte, and intercalary meristematic zone and foot. Okay, seta is absent over here. Seta is absent in the sporophyte. Look into it carefully. Gametophytic structure is just thalloid with rhizoids, and over this thalloid gametophyte, sporophytic structures are arising out. This sporophyte consists of what? Capsule, intercalary meristematic zone and foot. Seta is absent. Inside the capsule you can see spores and pseudoelaters. Pseudoelaters will help in easy dispersal of spores. Remember that, okay. Best example is what ma? Anthoceras, which belongs to Anthoceratopsida. Anthoceratopsida includes what? Hornworts. What is the gametophytic structure? This is the gametophytic structure. Over the gametophytic structure, the sporophytic structure will form. This is the gametophytic structure. Over the gametophytic structure, the sporophytic structure will form. Sporophyte in hornworts will just have capsule, meristematic zone and foot. Seta is absent. Capsule again has spores and pseudoelaters. Example is Anthoceros. Clear, right ma? Now, mosses you see. Very important. Liverworts are done. Hornworts are done. Now, mosses. Many a times asked question in your EAPCET from especially this segment of bryophytes. Remember carefully, here in mosses, gametophyte has two stages. First is primary gametophyte in the juvenile stage, okay. In the juvenile stage, the primary gametophyte which is formed is called as protonema. This is primary gametophyte. Primary gametophyte is protonema, okay, and this matures to adult leafy stage gametophyte. Adult leafy stage gametophyte is gametophore. How you can describe this protonema? Protonema is well described as creeping, green, branched, frequently filamentous stage, okay. This is described as creeping, green, branched, frequently filamentous stage. That is protonema, and this is the diagram of protonema, creeping, green, branched, frequently filamentous stage. So, here this develops directly what? This develops directly from the spore, okay. The haploid spore which is landing onto the substrata will first develop into the primary gametophytic structure, that is protonema. Protonema then matures to what? Leafy stage. How is it maturing? By lateral budding. Lateral branches of protonema will undergo budding and they develop into what? Adult leafy stage, adult leafy gametophytic stage. So, here what is adult leafy gametophytic stage? The upright slender axis which bears spirally arranged leaves. This is the upright slender axis which bears spirally arranged leaves. This is called as what? Leafy stage. So, here in the leafy stage you can observe rhizoids, main axis and leaves. All these are haploid. Gametophyte is haploid, rhizoids, main axis and leaves are haploid. Rhizoids here are multicellular rhizoids, okay, multicellular rhizoids. Multicellular rhizoids, clear, right? So, the apex has 6 organs, that is antheridia and archegonia, clear, right? So, in mosses you have gametophytic structures in two stages. One is juvenile stage and the second is adult leafy stage. Juvenile stage will develop into what? Juvenile stage is the primary gametophytic structure, that is protonema, which is creeping, green, branched, frequently filamentous structure. This undergoes lateral branching. Lateral branching will produce a leafy gametophytic stage, that is gametophore. Leafy gametophytic stage will have rhizoids, main axis and leaves, whose ploidy is haploid, and rhizoids of liverworts are unicellular, rhizoids of mosses are multicellular. Till here it is clear, ma? Yeah. Next you see, so the multicellular uniseriate filamentous sterile hair-like structures are present over the gametophyte. Those multicellular uniseriate hair-like structures are called as paraphysis. Remember. So, from this gametophytic structure, sporophyte will arise. Okay, from this gametophytic structure, what is arising? Sporophyte is arising. This sporophyte is having capsule, seta and foot. Okay, it has capsule, seta and foot. Sporophyte is diploid and it has what? Capsule, seta and foot. Foot, seta, capsule are present. So, this sporophytic structure is more elaborate than that you see in the liverworts. Capsule at the tip. Capsule has what? Spore mother cells. Of course, all the sporophytic structures are what, ma? diploid. Here you have to remember the ploidy. Okay, we know in bryophytes the dominant gametophytic structure. What is the dominant plant body? Gametophytic plant body is dominant. Dominant plant body. So this dominant plant body is what ma? Haploid. It has rhizoids, main axis and leaves. Rhizoids, main axis and leaves, ploidy is haploid. From the gametophyte, sporophyte will arise. This is dependent on the gametophyte as partial parasite. This is nutritionally dependent on the gametophyte. Okay, sporophyte ploidy is diploid. Okay, it has capsule which is diploid, seta is diploid, inside that you have foot. Foot, seta and capsule. In capsule you have what ma? Spore mother cells. Spore mother cell is diploid, which undergoes reductional division or meiosis, which is called as sporic meiosis. Sporic meiosis forms 4 haploid spores. 4 haploid spores. Fine, 4 haploid spores. Like how you have observed elaters which aid in the dispersal of spores or dispersal of antherozoids from antheridiophore, here you have peristomic teeth which aid in the dispersal of spores. Peristomic teeth which is present in the capsule, peristomic teeth which is present in the capsule will aid in the dispersal of spores. Okay, and there it is elaters, here it is peristomic teeth which aids in the spore dispersal. Till here it is clear, no ma? Next you see. Yeah, Funaria, best examples of mosses and bryophytes include Funaria, cod moss, Sphagnum, peat moss and Polytrichum, hair cap moss. Sphagnum, if you see in the same plant body, you can see antheridial branch and archegonial branch. So, this is what? Heterothalic or bisexual. Bisexual or monoecious. Sphagnum is bisexual or monoecious. Okay. Whereas Polytrichum and Funaria both are dioecious or unisexual. Clear, right? Important ma, you have to remember. Yeah, next. Look into the economic importance of bryophytes. Economic importance of bryophytes, if you have to see, mosses will play major role in plant succession. Of course, the pioneers of plant succession are what? Lichens. Okay, lichens are the starters of the plant succession, where we popularly call it as what? Pioneers of plant succession. Plant succession. Fine, right ma? So, mosses play important role in plant succession. This prevention of soil erosion, like sphagnum is also called as peat moss, bog moss, cotton moss. Very important, n number of times asked question on the economic importance of sphagnum. It is also called as peat moss because it is well grown in the marshy areas and it is used as a good manure and fuel. This is called as peat moss. And sphagnum is called as bog moss because it builds soil in rock crevices, it will fill ponds and lakes. Okay. This is also called as cotton moss because this dry sphagnum is used for the transshipment or transportation of seedlings and cut plants. Okay. This is used in the transportation of seedlings and cut plants. Sphagnum also called as peat moss, bog moss, cotton moss. Peat moss because it can grow in the marshy areas and it is used as good manure and fuel. Bog moss because it will fill the rock crevices and it will fill the ponds and lakes. Cotton moss because it absorbs the water and it helps in the transshipment of commercially important plants. Okay, clear ma? You have understood why is it called as peat moss, bog moss and cotton moss. This is the economic importance of bryophytes. And then life cycle, very important. I already told you, not just in EAPCET, just in NEET 2025 paper, you got a multi-statement question on the stages of life cycle both in bryophytes and pteridophytes. So, look at it carefully beta. What type of life cycle does bryophyte has? Of course, the main or dominant plant body is gametophytic plant body only. It exhibits haplodiplontic life cycle. Haplodiplontic life cycle. Okay, haplodiplontic life cycle. So, here if you see, first let us start with the gametophytic structure. So, the primary gametophytic structure is protonema, which is green, creeping and branched. This protonema through lateral branching develops into a main gametophytic structure. This has an axis, rhizoids and leaf-like structures, and this has both male gametophyte and female gametophyte. Male gametophyte has antheridia, which is covered by sterile jacket layer. Female gametophyte has archegonia, which has egg cell or oosphere at the base. Antheridia will produce out antherozoids. These antherozoids are released out by the help of elaters in liverworts and with the help of peristomic teeth in the mosses. After the release of antherozoids, what happens? This fertilizes with the egg. Both haploid antherozoid fertilizes with haploid egg, and post fertilization what is formed? Diploid zygote is formed. Post fertilization diploid zygote is formed, right? And here site of fertilization or location of fertilization is what ma? Archegonium is the location of fertilization. This is diploid zygote, matures to diploid embryo, and this develops to diploid sporophyte. This develops to what? Diploid sporophyte. And this diploid sporophyte consists of what ma? If it is funaria, it has foot, seta and capsule. Foot, seta and capsule all are diploid only. So, inside the diploid capsule you have sporogenous tissue, and this sporogenous tissue has sporogenous cells which are called as spore mother cells. Spore mother cells are diploid. This spore mother cells inside capsule, they will undergo what ma? Sporic meiosis. Spore mother cells undergo sporic meiosis, forming haploid spores. Okay, forming haploid spores. These haploid spores are released out with the help of peristomic teeth, and the spores are what? Ploidy of the spores is haploid spores. Haploid spores landing onto the suitable substrata, they will develop into a new gametophytic structure, that is protonema. Clear, right ma? So, here it exhibits equal haplontic phases and equal diplontic phases. So, what is the life cycle? Haplodiplontic life cycle, and the major or dominant plant body is gametophyte. This thing you have to remember. Sporophyte is dependent on gametophyte. How is it dependent? Nutritionally it is dependent for nourishment. Till here it is clear, right ma? So, bryophyte is done. Now, we will move on to pteridophytes. In pteridophytes, first you have to observe the general characteristics again. Okay, general characteristics of pteridophytes. These are the first terrestrial land plants which are vascular. First non-vascular terrestrial plants are bryophytes. First terrestrial vascular land plants are what? Pteridophytes. And how can you describe it? These are the embryophytic but not first embryophytes. First embryophytes are bryophytes, and these are archegoniate. First archegoniate, again bryophytes. Tracheophytic. Bryophytes are atracheophytic, pteridophytes are tracheophytic because they contain tracheates. Tracheates as their conducting elements. Okay, and they are seedless vascular plants. Remember the description clearly. Okay. Now, when you look into the habitat is cool, damp, shady places and sandy, soily conditions. These are the this is the habitat of pteridophytes. If you look into bryophytes, habitat is cool, damp and shady places. Here it is cool, damp, shady and soily areas also. And here main or dominant plant body is sporophyte. There in bryophytes the dominant plant body is gametophyte. Here in pteridophytes the dominant plant body is sporophyte. Sporophyte is independent of gametophyte here. Okay, there in bryophyte, sporophyte is dependent on gametophyte. In pteridophyte, sporophyte is independent of gametophyte. Here both gametophyte and sporophyte both are independent to each other. Remember, very important statement. In pteridophytes, gametophyte and sporophyte both are independent to each other. Okay, and sporophyte is independent to gametophyte. Clear right now? This includes club mosses, horsetails and ferns. Next, if you see main plant body is sporophyte. This is differentiated into root, stem and leaf. In bryophytes though, you have root like, stem like, leaf like structures, whereas in pteridophytes you can see root, stem and leaves. Roots are adventitious, leaves are small, microphyllous, large, macrophyllous. Here leaves are called as sporophylls beta. Sporophylls. Leaves are addressed as sporophylls because leaves contain sporangia. They consist of what? Sporangia. They are called as sporophylls. Sporangia, they are called as what? Sporophylls. Leaves are small microphylls and large macrophylls. Small microphylls leaves you can see in Selaginella, large macrophylls you can see in ferns. Clear, right? Next, stem. Internal anatomy of stem shows epidermis, cortex and stele. And stele is very simple, primitive stele, that is protostele. Protostele, where at the center xylem will be there. Xylem is surrounded by phloem. Okay, this is the primitive stele, that is protostele. Next, leaves and leaflets, as I told you in pteridophytes, they are bearing what? They bear the sporangia. That is the reason they are called as sporophylls. Leaves and leaflets, here they are addressed as sporophylls. How sporangia is developed on the leaves? If sporangia is developed from the single cell, if single cell develops into sporangia, that is called as leptosporangiate. Okay, if single cell develops into sporangia, that is called as leptosporangiate. If group of cells develop into sporangia, that is termed as eusporangiate. Single cell develops into sporangia, termed as leptosporangiate. Group of cells developing into sporangia known as eusporangiate. So here sporangia or spore mother cell, which is diploid, undergoes sporic meiosis again. Sporic meiosis. If you notice the evolution carefully, just a second ma. Sporic meiosis. Yeah, if you notice the evolution carefully, you can observe zygotic meiosis in algae, sporic meiosis in bryophytes and pteridophytes. Same the case here in pteridophytes. Sporangia which are present in the leaves and leaflets. How sporangia is developing? Through leptosporangiate, eusporangiate. Leptosporangiate single cell develops into sporangium, eusporangiate group of cells will develop into sporangium. Okay. So here the diploid sporangia undergoes sporic meiosis, forming out what, Nana? Haploid spores. This haploid spores can be homosporous or heterosporous. Remember, first heterosporous condition is observed in pteridophytes only. First heterosporous condition is observed in pteridophytes only. Homosporous, purely homosporous condition is observed in bryophytes. First heterosporous condition is observed in pteridophytes, wherein you can see two types of spores, microspores and megaspores. So the heterosporous condition, you can take two best examples, Selaginella and Salvinia. Along with this, you can take other two examples also, Marsilia and Azolla. These are the best examples of heterosporous condition. Means these pteridophytes will produce two types of spores, microspores and megaspores. Microspores will develop into female reproductive structure, megaspores into male. Clear, right? Microspores develop into female gametophytic structures, microspores develops into male gametophytic structures, megaspores develop into female gametophytic structures. Clear, right ma? Clear? You have understood how sporophylls are present, how sporangia is developing on the sporophylls, and this sporangia has what type of cells? Spore mother cells. Undergoes what meiosis? Sporic meiosis. Produces what spores? Haploid spores. Haploid spores are of two types. What are they? Homosporous and heterosporous condition. In heterosporous condition, microspores matures to what? Male gametophyte. Megaspores matures to what? Female gametophyte. Clear till here, no? Any doubt anywhere? And the examples of heterosporous condition is very important. This statement, fix it in your mind. First heterosporous condition is observed in what? Pteridophytes. Next, homosporous condition where gametophyte is monoecious. Okay, heterosporous condition where gametophyte is dioecious. Okay, gametophyte is dioecious. Dioecious or unisexual, remember. Gametophyte is monoecious, which means bisexual, excuse me. Gametophyte is dioecious, which is unisexual. Means both male and female gametophytes are separate here. Here they are present in the same plant body. Now here you see, in few plants like Selaginella and Equisetum, you have strobili or cone. Okay, strobili or cones are seen, which are at the apex of the pteridophytic plant bodies. At the apex you can see strobili or cones, where sporophylls will form compact structures. Distinct compact structures are observed. For example, Selaginella and Equisetum. Okay, strobili or cones where sporophylls form distinct compact structures. For example, Selaginella and Equisetum. Clear, right ma? You have to remember the examples for sure. There is no other option at all. You have to remember. Okay, so spores will germinate into what? There in the bryophytes, the primary gametophytic structure is protonema. Here in pteridophytes, the primary gametophytic structure is prothallus. Prothallus is the primary gametophytic structure. So spores first germinate into prothallus. Prothallus, they are small, inconspicuous, multicellular, free-living photosynthetic thalloid gametophyte. This is the description of prothallus. Their protonema you can describe as what ma? Green creeping branched structure. Here it is small, inconspicuous, multicellular, free-living photosynthetic thalloid gametophyte. This is the description of what? Prothallus, remember. Okay, and this will develop into what? Prothallus will mature into mature gametophytic structure which bears sex organs. Sex organs, again here they are multicellular, jacketed and sessile sex organs. Heterosporous condition you can observe, I already told you, megaspores will develop into female gametophyte, microspores will develop into male gametophyte. Next ma, this is the life cycle of pteridophytes, wherein first we will start with gametophyte. Here haploid spores undergoing mitosis forms primary gametophytic structure. That primary gametophytic structure is prothallus. Prothallus is the primary gametophytic structure. This develops into mature gametophytic structure which is of two types. It can be male gametophyte and female gametophyte. Monoecious it can be or dioecious. After fertilization, what is the site of fertilization again? Site of fertilization is archegonia and mode of fertilization is what? Zooidogamous oogamy. Same zooidogamous oogamy you can find in bryophytes. Fertilization, diploid zygote is formed. Diploid zygote will mature to embryo and embryo is present inside the female gametophyte for variable periods of time. For variable periods of time. Okay, embryo is retained for variable periods of time inside the female gametophyte. Okay, inside the female gametophyte, embryo is retained for variable periods of time, but it is not completely retained because of the absence of integuments. It is not completely retained because of what ma? Absence of integument. So here embryo retaining for variable periods of time, this is termed as what nana? Precursor to seed habit. Okay, two peculiar features specific features that you have to remember with respect to pteridophytes is, first is heterosporous condition, second is precursor to seed habit. These are two important features that you have to keep it in your mind with respect to pteridophytes. Now, this is a precursor to seed habit. This is a very important step in the evolution. This marks as a major important step in the evolution, and this develops into a mature sporophyte. Sporophyte, you can say this is the multicellular, well differentiated dominant phase, okay, whereas in bryophytes that is the gametophyte is the dominant phase. In pteridophytes, sporophyte is the dominant phase. Clear ma? Any doubts anywhere? Clear, right? What are the two specific features of pteridophytes? One is heterosporous condition, second is precursor to seed habit. Pteridophytes are classified into 4 classes. Bryophytes, there are classified into 3 classes, right? Hepaticopsida, Anthoceratopsida, and one more thing is mosses. Okay. Here pteridophytes are classified into 4 classes. One is Coelopsida, second is Lycopsida, third is Sphenopsida, fourth is Pteropsida. So here you see ma, Coelopsida, best example is Coelotum. Lycopsida, it is Selaginella and Lycopodium. Sphenopsida, Equisetum. Pteropsida, Bryopteris, Pteris and Adiantum. These are the examples you have to remember for sure. Quite a times in your EAPCET PYQ, directly questions are asked from the classification of pteridophytes. Okay, 4 classes, in each class it's examples. Okay. Next is Dryopteris, important for EAPCET. So here you see the stem here has stem here in Dryopteris is underground rhizome. It is modified to underground rhizome. Leaves are large, aerial, and it is pinnately compound. They are addressed as fronds. Petioles, you can see petioles, those structures which bear leaves, they are covered by multicellular hair-like structures called as ramenta. Leaflets, they open dichotomously. Leaflets open dichotomously, you can see here, and this exhibits furcate venation. Sporangia is formed on the ventral surface or adaxial surface. Over the ventral surface, what are present? Sporangia are present. This groups to sori, okay, and in this sporangia only spore mother cells are present which undergo sporic meiosis. Antherozoids are coiled and multiciliate. These are the characteristic features of what? Pteridophytes. Fine. This is in extra with NEET. For NEET aspirants, this is not required. Okay. Only for EAPCET aspirants. This slide belongs to EAPCET aspirants. Okay. Next, each sorus is covered by membranous sheet. Each sorus is covered by membranous sheet, that is called as true indusium, and then it is pteris. Pteris, each sorus is like this, which is protected by reflexed margin of fertile leaflet. Each sorus is protected by reflexed margin of fertile leaflet. This is false indusium. Okay. Here you can see in Dryopteris true indusium, in Pteris it is false indusium. Economic importance of pteridophytes. This is the last slide of today's session. Beta. Economic importance includes pteridophytes are used as soil binders. Second, these are used in the medicinal purposes as well, wherein Dryopteris is bestly used as what? Anti-helminthic drug. And equisetum stems are used in soaring, polishing of metals. Equisetum stems are used in what ma? Soaring, polishing of metals. These are the key features of pteridophytes. What all we have discussed in today's session are very important. Bryophytes and pteridophytes are two major important domains of this chapter, plant kingdom. Every year definitely you will get a question from bryophytes and pteridophytes. There is no EAPCET paper which is escaped without getting a question from bryophytes and pteridophytes. That is the reason in today's session I have dealt only bryophytes and pteridophytes. Tomorrow in our next session, when we are solving EAPCET PYQs, you will get to know how important this class is. Okay. So, come prepared with algae, bryophytes, pteridophytes and methods and types of classification of plant kingdom and branches of taxonomy. Tomorrow we will deal with gymnosperms and angiosperms, and then we will solve EAPCET PYQs. Once again, I would like to end up the session by telling you how important this playlist is for all the EAPCET aspirants. For all the EAPCET aspirants who want to crack EAPCET 2026, complete content with EAPCET PYQs are available with us. Make use of this public platform, and that's all for today's session. Thank you.